Welcome you all to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test MSc Entrance question paper Biochemistry conducted in the year 15th June 2023. I have discussed only the biochemistry portion. If you are new to my channel, please do like, share and subscribe. If you want still previous year's question papers of biochemistry, I have uploaded them in my channel Omni Bio Entrance, and you can get them from the channel playlist. For coming to the first question, identify the correct statement about homotropic. Now what is homotropic enzyme regulation? Now allosteric regulation, there are two types on the basis of substrate and effector molecule. One is homotropic regulation and the other is heterotropic regulation. In homotropic regulation, substrate molecule act as an effector also. That is, example is binding of oxygen to hemoglobin molecule. And in heterotropic regulation, substrate and effector are different. The effector can either activate or inhibit the enzyme. That is in the case of heterotropic regulation. So here they have told identify the correct statement about homotropic. So for regulatory enzyme, substrate and modulator are identical. Answer is option 1. Modulator is a molecule other than the substrate. That is in the case of heterotropic regulation, not homotropic. The other two options are not related to homotropic and heterotropic. Then next one. Diisopropyl phosphofluoridate DIPF act as an inhibitor of enzyme with serine at active site. So it's an inhibitor of enzyme which has got serine at the active site. Then type 4 delayed hypersensitivity is not caused by. So type 4 hypersensitivity, also known as delayed type hypersensitivity, is a cell-mediated immune response that occurs after about 24 to 72 hours of exposure to the antigen, and it is characterized by activation of T cells, macrophages and release of lymphokines. So first option, macrophage, it is involved in delayed hypersensitivity. Lymphokines are also involved. Circulating antibodies are not involved, and they have asked which is not caused. So circulating antibodies is the answer. Then next one. Which of the following statement is correct about enzyme? So you have to find out the correct statement about an enzyme. A biocatalyst that increases the rate of chemical reaction. That statement is correct. That is your answer. A biocatalyst that inhibits the rate of chemical reaction. No, it increases the rate of chemical reaction. Non-specific in nature. Enzyme substrate reactions are specific in nature. All enzymes are proteinaceous in nature. No, there are ribozymes which are RNA molecules. Then oxidases act as. So oxidases transfer electron from substrate to oxygen, resulting in the formation of water or H2O2. Then there are others which are called as oxygenases. They incorporate oxygen into the substrate molecule, whereas oxidase do not incorporate oxygen into the substrate molecule. So first statement, use oxygen as an electron acceptor but do not incorporate it into the substrate. That is oxidases. Directly incorporate oxygen into the substrate, that is oxygenases. So first statement is the correct answer. Then next one. Enantiomers are special type of stereoisomers that are. So enantiomers, they are mirror images of each other. So the answer here is option 4. Then next one. Which of the following is the biologically active form of vitamin D that regulates calcium levels in the kidney and bone? So biologically active form of vitamin D, we all know it is called calciferol. So first option they have told dehydrocholesterol, that is not correct. Then next three options, there is called calciferol in them. But the most apt one will be 1-alpha 25 dihydroxycholecalciferol. Answer is option 3. Then next one. Biological value of a protein can be defined as. So biological value is nitrogen retained by nitrogen absorbed into 100. First option is the answer. That is amount of nitrogen retained in the body by the amount of nitrogen absorbed from the protein. That is called as biological value of proteins or BV. Then next one. Which of the following parameter is used to assess the rise in insulin following consumption of any food? So it is glycemic index, option 3. And glycemic index, it is the measure of how quickly a food raises blood glucose levels, which is then triggers an insulin response. That is called as glycemic index. And foods with high glycemic index cause rapid increase in the blood glucose, resulting in greater insulin response. Now PADCAS, that is your option 1, that is protein digestibility corrected amino acid score. It evaluates protein quality and digestibility. Then nitrogen balance, it assesses protein metabolism and balance in the body. Then glycogen index, it is not recognized in any nutrition science. So the answer here is glycemic index. Then next one. Water can dissolve many organic molecules due to. So this question they have been asking from your NEET examination. So it is dipolar character and ability to form hydrogen bonds. Answer is option 1. Then cellulose is an unbranched polymer of glucose residues joined by. So glucose residues in cellulose, they are joined by beta-1,4-glycosidic linkages. Answer is option 3. Then next one. A person having a BMI, body mass index, of 35 will be recognized as. So as per the WHO BMI classification, he or she will be recognized as grade 2 clinical obesity. Answer is option 3. Then which of the following additives need to be added to the blood sample to prepare serum for any chemical analysis? Now serum is prepared by allowing the blood to clot and then centrifugation it and then separating the solid components from the liquid serum. So no additives are usually needed to do this procedure. The citrate, oxalate, EDTA, they are all anticoagulants used to prevent blood clotting. So the answer here is option 4. Sorry. Then next one. Prostaglandins are synthesized in vivo from the 20-carbon polyunsaturated fatty acid. So which fatty acid is involved in prostaglandin synthesis? So it is arachidonic acid. Answer is option 2. Then next one. Creatinine is used to assess the renal glomerular function because creatinine is easy to estimate. No, the value of creatinine clearance is close to GFR. That is correct. GFR is glomerular filtration rate. So the value of creatinine clearance, that is close to GFR. That is the answer. A 20-year-old boy was advised a liver function test by the doctor upon complaining a pain in the abdominal region and yellowing of nails. So a 20-year boy was advised liver function test because he complained of pain in the abdominal region and yellowing of the nails was observed. So the doctor advised him for a liver function test. Subsequently, he was diagnosed of hepatic jaundice. So, after all the tests and analysis of the test result, he was diagnosed of hepatic jaundice. Which of the following would you expect in his test report? Increased activity of ALT and AST. That is the right answer. Now what is ALT? AST. ALT is alanine transaminase and it is an enzyme which is primarily found in the liver, and AST is aspartate transaminase and it is found both in the liver and the muscles. In hepatic jaundice, liver damage leads to elevated levels of ALT, that is alanine transaminase, and moderately elevated levels of AST, that is aspartate transaminase. Both these get elevated because of liver damage. So the answer here is option 1, increased activity of ALT and AST. Then next one, Niemann-Pick disease is caused by due to genetic deficiency of. So Niemann-Pick disease, it is a group of genetic disorders that are affecting lipid metabolism. So it is caused due to genetic deficiency of sphingomyelinase. Answer is option 3. The next one, the condition fatal infantile mitochondrial myopathy and renal dysfunction involves presence of fumarate, absence of most oxidoreductases of respiratory chain. Yes, that is correct. And the oxidoreductase is usually the cytochrome C oxidase, which becomes deficient in fatal infantile mitochondrial myopathy and renal dysfunction. So the answer here is option 2. Then next one, which is not a part of the cytoskeleton? Serpin, tubulin, actin, keratin. It is not keratin, it is a printing mistake, it is keratin. So which is not a part of cytoskeleton? It is serpin. Cytoskeleton, you all know there are 3 types of protein fibers in the cytoskeleton, that is microfilament, intermediate filament and microtubules. The microfilaments, they are made up of actin, intermediate filaments are made of keratin and microtubules are made of tubulin. So they have asked which is not a part of cytoskeleton, so it is serpin. Then next one. Cyanide is probably the most potent inhibitor of electron transport chain. It binds to cytochrome oxidase. So specifically Fe3 plus of cytochrome oxidase. Answer is option 4. Then next one. Mass to charge ratio or M by Z ratio can be measured through. So mass to charge ratio or M by Z ratio, it is measured using a mass spectrophotometer. It measures the mass to charge ratio of ions and it provides information on molecular weight, structural fragments and isotopic composition. Whereas gas chromatography, it separates the molecule based on boiling point and affinity for stationary phase. UV visible spectrophotometer measures absorption of light by molecules. Fluorescent spectrophotometer, it measures the emission of light by excited molecules. So they all do, these first three options, they do not consider any mass to charge ratio. So it is only the mass spectrophotometer which will consider the mass to charge ratio. Then next one. Which of the following organelle is pelleted first during subcellular fractionation by differential centrifugation at 1000 G for 10 minutes? So using differential centrifugation, which organelle will be pelleted first at 1000 G for 10 minutes? So it will be the most densest organelle at such a low speed. because densest and the largest. So the answer here is nuclei because it is densest of the organelle and size is also relatively large and it will sediment faster or for at first. Then next one, another name for cardiolipin is, so it is diphosphatidyl Answer is option 4. Then next one, which compound in red cells is crucial in determining the oxygen affinity of hemoglobin? So it is 2,3-bis phosphoglycerate. 2,3 it binds to hemoglobin reducing its oxygen affinity. 2,3-bis phosphoglycerate, it will bind to hemoglobin, reduce its oxygen affinity, then and it facilitates the release of oxygen to the tissues. That is what 2,3-bis phosphoglycerate does. Moreover, it shifts the oxygen hemoglobin dissociation curve to the right. And from where does the red cells get 2,3-bis phosphoglycerate? That is produced from glycolysis and glycolysis. Then inositol triphosphate. Option 1, it is involved in cell signaling. Glyceraldehyde 3-phosphate, it is an intermediate of glycolysis but it doesn't have anything to do with oxygen affinity of hemoglobin. Then phosphatidyl inositol, it is a component of cell membrane and it also doesn't have anything to do with oxygen affinity of hemoglobin. Then next one, proteins can be separated electrophoretically on the basis of net acidic and basic charge through. So on the basis of charge, proteins can be separated by isoelectric focusing. Then next one, molarity. What is molarity? It is number of moles of solute in 1 liter of the solution. Answer is option 1. Then next one, synthesis of 2,3-bis phosphoglycerate increases in high altitude because. So I already told 2,3-bis phosphoglycerate, it binds to hemoglobin reducing its oxygen affinity and thereby facilitate the release of oxygen to the tissue. So what does 2,3-bis phosphoglycerate does? 2,3-bis phosphoglycerate favors the relaxed state of hemoglobin. No. 2,3-bis phosphoglycerate serves as an energy source at high altitude. No. It is involved in oxygen affinity for hemoglobin. So 2,3-bis phosphoglycerate stabilizes the T conformation of hemoglobin. Yes. Thereby decreasing the oxygen affinity of hemoglobin. Yes. And making it more available to the peripheral tissues. That is correct answer. Answer is option 3. Then next one, the drug sildenafil or Viagra works by. So what it does is it relaxes the smooth muscle and vasodilation by inhibiting cGMP phosphodiesterase. Answer is option 3. Then next one, in the pump Na plus K plus ATPase during active transport, hydrolysis of ATP provides energy for. So how many moles of Na plus will go out, how many of the K plus will enter into the cell, you should know for Na plus K plus ATPase. So it is 3 moles of Na plus out of the cell and 2 moles of K plus into the cell. Answer is option 3. Then next one, inspiratory capacity can be calculated as tidal volume plus inspiratory reserve volume. Answer is option 1.